Hello everyone, I'm Dr. Devi, Associate Professor, Department of Genetic Engineering, SRM Institute of Science and Technology. I'm here today to explain and demonstrate another important experiment of Cancer Genetics Laboratory, Immunofluorescence assay of cancer biomarkers. Immunofluorescence is a powerful technique in biology and it is used to detect specific proteins or antigens in biological samples. The biological samples can be tissues or cells. Immunofluorescence relies on specific antibodies which are used to detect specific proteins which are present in the cells using fluorescence microscopy. Biomarkers are molecules which are indicators of a disease condition of a patient. Cancer biomarkers are molecules which are secreted by the cancer cells in the body and they indicate the presence of cancer in the body. Today we are going to identify a protein cancer biomarker in the HeLa cells by immunofluorescence. Immunofluorescence technique involves a number of steps and today we are going to see step-by-step procedure of the entire experiment. As we are going to detect the presence of a protein cancer biomarker in HeLa cells, the first thing we have to do is to culture the HeLa cells. Culturing HeLa cells has to be done on cover slips placed on Petri plates because it is easy to mount them and to visualize them using fluorescent microscope. The cover slips have to be sterilized before culturing the HeLa cells and they have to be sterilized under UV for at least 4 hours. After sterilization of the cover slips, the HeLa cells have to be seeded onto these cover slips which are placed on the Petri plates and they have to be allowed to grow in the incubator. Once they are confluent, the Petri plates have to be taken out of the incubator and the media has to be removed and the cells have to be first fixed. Fixation is an important aspect of immunofluorescence as this fixation helps to preserve the cellular morphology and it also prevents degradation or denaturation of the cells. Fixation of the cells can be done using 4% paraformaldehyde for about 10 to 20 minutes at room temperature, and after fixation the fixative has to be removed and the cells have to be washed with ice cold PBS. The next step after fixation is permeabilization and this has to be done especially if you are planning to visualize or detect proteins which are intracellular. Permeabilization helps in opening up the pores of the membranes and the usual permeabilizing agent which we use is Triton X100. 0.1% of Triton X100 can be used for permeabilization and it is done for about 10 minutes. After permeabilization, the Triton X is removed and the cells are washed with PBS 3 times with a duration of about 5 minutes for each wash. Now we have completed the first 2 steps, fixation and permeabilization in the immunofluorescence technique. As you all know, immunofluorescence relies on specific antibodies which bind to specific target epitopes on the antigens. There are 2 types of immunofluorescence, direct immunofluorescence and indirect immunofluorescence. In the direct immunofluorescence, the primary antibody is conjugated to a fluorophore which binds to the target epitope on the antigen. And this can be visualized using the fluorescence microscope. The next step in immunofluorescence is blocking and this is done to prevent the antibodies from binding to non-specific epitopes which are present. The blocking buffers or reagents which are used are bovine serum albumin or skimmed milk. Today we are going to use 1% BSA for blocking. Blocking can be done at room temperature for about 1 hour depending upon the cell type, and once blocking is done, the blocking reagent is removed and the cells are again washed with PBS, and during the washing procedure PBS can also be combined with Tween 20 and PBS-T buffer can be used for washing the cells. And every time when you're washing the cells, the wash has to be done for 5 minutes 3 times using the PBST buffer. The next step after blocking is incubation of the cells with the primary antibody. So as the blocking is being done, the primary antibody can be diluted. Today we are going to use 1:100 dilution of the primary antibody in the blocking buffer. And after blocking is over, the cells can be incubated with the primary antibody at 4 degrees centigrade overnight. Or it can also be incubated at room temperature for 1 hour. We actually prefer to do it at 4 degrees centigrade overnight. The next day, the primary antibody is removed from the cells and the cells are again washed with PBST. After this, the cells are incubated with secondary antibody at room temperature for about 2 hours. The secondary antibody is also diluted at the ratio of 1:200 dilution, and it is diluted in the blocking buffer. The incubation of the secondary antibody has to be done in the dark because it is conjugated with the fluorophore, and the antibody which we are using is conjugated to FITC or fluorescent isothiocyanate. After incubation with the secondary antibody, the cells are washed again with PBST, and at the same time we can also counterstain the cells with DAPI, which is a nuclear stain. After incubating the cells with DAPI for about 5 minutes, it is removed and the cells are again washed with PBST, and the cells are air dried and the cover slip is slowly removed from the Petri plate, and it is now ready for mounting onto the slides. Mounting can be done using glycerol or any other suitable mounting medium, and care has to be taken to make sure that the cells are mounted, or the cover slip is mounted with face down so that the cells are onto the slides, and this would make easy observation of the cells under the fluorescent microscope. Now we are visualizing the cells in the fluorescent microscope, and we can observe the presence of the protein cancer biomarker in the nuclear region of the cells. When visualizing under the fluorescence microscope, you can detect and localize the presence of the protein cancer biomarker in the nuclear region, which is an indication that it is a nuclear protein. And you can see that this nuclear protein is upregulated or present in the HeLa cells, which is a non-metastatic cervical cancer cell line. The usual steps which is followed for immunofluorescence, and hope you have enjoyed the video. Thank you.